everyone. We have had a really exciting morning and a full lunch. And I'm going to give you a talk on evolution theory. So now to fight off any potential sleepiness or fear for mathematics, I have reduced the number of slides from 30 to 8. Because 8 is a lucky number in the Chinese culture. And I need it, because I'm experimentalist, and this is the first time I will give a pure theory talk. So in fact, I'm a bit nervous. So now I want to open my talk by posing a question. Suppose I have a community of 6 bacterial species. It does something interesting. For example, it can convert cellulose, an agricultural waste, to a useful product, such as an anti-cancer drug. However, the activity is a bit low, and I want you to help me to improve this activity. What would you do? You might say, OK, the first thing I want to do is to figure out whether a single species can do the job. Because if it can, then I won't need to worry about the 5 other species, right? So we'll go back to the lab, we isolate single species, we check for its activity. No single species has that activity. So this is what I define as community function, a biochemical activity that can be performed by a community, but not by member species alone. Now next, you might want to figure out what is the minimal subset of species required for this activity. So you will do standard dropout experiment. You throw out this species, you throw out that species, so on and so forth. At the end of the day, you congratulate yourself because you're very lucky. Only 2 species are required for this activity or function. Now what would you do? How would you improve the community function of converting the cellulose to the anti-cancer drug? So one way to do this is to figure out how these 2 species interact, how they work together to have this activity. Then you will need to figure out how A affects B and how B affects A. And this is by no means a trivial task. This is because each species can release tens, if not hundreds of compounds. And many of these compounds can elicit complex physiological reactions in the partner species. And now you have, suppose you could, in fact, compile this haystack of interactions. You would still need to figure out from this haystack which are the needle interactions that are truly critical for the community function of interest. And suppose you could, and then you will need to modify these 2 species or their abiotic environment. You tweak this knob, you tweak that knob in the hope that you would be able to improve community function. This all sounds quite complicated, right? But on the other hand, you could say, why not try an artificial selection experiment? That is, we grow up many, many copies of this community, allow mutations to accumulate. And then we artificially select those communities that have the highest function. And then we perform assisted reproduction on these communities in the sense that we would take these high performers and let them reproduce by splitting each into newborn communities, baby communities, and allow them to grow again into adults. And this cycle can repeat itself. The conventional wisdom says you get what you select for. But is this true? I want to tell you that it is often not true. But if you are smart about doing the experiment, selecting communities based on their functions could work. Now, before I dive into details, I want to take one step back and consider selection on individual cells. Furthermore, I want to contrast natural selection versus artificial selection. So suppose you have a population of cells. You want the fastest growing cells. What would you do? You would take cells, put in the test tube, and let them grow up. You would take a subculture and go to the fresh tube. You repeat this process. The fastest growers will always take over, and that's natural selection. The survival of the fittest, in this case, the survival of the fastest growers. Now imagine this population of cells are expressing GFP, and what you are interested in is the brightest cells. And note that brightest cells are very likely not to be the fastest growing cells, right? Because expressing GFP inflicts a fitness cost. So what would you do instead is performing an artificial selection experiment. You would, for example, grow a population up. You would artificially select for these brightest ones using a machine called flow sorter, and sort these brightest cells to a fresh test tube and allow them, and only them, to grow up. And you repeat this process. And this, in fact, has been done very successfully. So for selection to work, you require 3 key elements: variation, selection, and heredity. Here, variation is caused by mutations. Mutations can introduce variations in genotypes and thus phenotypes among individual cells. Selection, that's artificial selection. It's done by us, not by nature. Third, you need heredity. And that is satisfied because bright cells from this cycle tends to be bright next cycle, saving for rare mutational events that would dim the GFP expression. So when all these 3 criteria are satisfied, we could have selection work for us. But how would these 3 elements play out if you are selecting from communities of multi-species microbial communities instead of individual cells? This experiment has in fact been performed only a few times, not many times, a few times. So I want to give you an example. In this experiment, the researchers are interested in identifying microbes that would degrade industrial pollutant. So they will go to pond, they isolate microbes from the pond and mix it up such that the starting point is more or less similar. Then they inoculate into tubes, and 15 of them. And I call them newborn communities. You might already be a bit upset, right? Because newborn is used to usually describe newborn individuals. But remember here, the unit of selection is not individual, but rather communities of cells. So this is artificially set up by experimenters. And they allow these communities to grow for 4 days, and I call that maturation time. And cells would grow, and we get 15 adult communities. And again, this is artificially chosen. So when I say adult, I do not necessarily mean that these communities have some specific physiological state, such as steady state ratio or saturation. This is all artificially done. So I sometimes would call newborn communities adult communities, just as newborns or adults. So then the researchers will pick the 3 highest activity communities. Those communities have degraded the most pollutant, and allow them to reproduce by randomly partitioning a community into multiple newborn communities so that you can have 15 newborn communities for the next cycle. This is what I call as a selection experiment. Now, as a control, the researchers randomly select 3 adults and reproduce by splitting. And this I call a control experiment. So out of 4 control experiments, community activity of degrading pollutant increased in 2 and decreased in 2. Out of 4 selection experiments, this activity went up in 3 and declined in 1. Obviously, the number of experiments is still small. But even if the experiment had worked in a statistically significant fashion, we still would not know the mechanisms. For example, we would not know whether pollutant degradation is a community function or whether it can be performed by a single species. Second of all, we do not know whether they are selecting from a subset of species or genotypes within species. And third, its most kind of subtle point, is that it's actually not clear whether this is actually just natural selection in disguise. For example, these microbes can now, for example, adapt to use this pollutant as carbon source. And as a result, they would just grow faster on this new carbon source. And this would be indicated as improved community activity. So it's actually not clear whether community selection per se is required for this kind of activity. So we want to look more at how we might do such tricky experiments. I remember going to an evolutionary biology meeting and asking 3 well-respected evolutionary biologists. I said, I want to do such an experiment, so tell me how many communities should I start from, should I select from? The first answer was, that is a great question. The second answer was tens of communities, just like this one. The third answer was millions of communities. Now, this drastically different answers is very discouraging to us as experimentalists. We're almost facing what's like the situation Trevor or the team have been talking about, right? Because I'm just asking them about one of the many experimental parameters that we need to be wary of. So that time we decided to drop experimental efforts, but instead doing some theory first to train our intuition about how to do this type of experiments. So by we, I mean Li Xie, a very talented postdoc in my lab, and I myself. And I want to make a disclaimer that from this point on, if I say experiments, I meant computational experiments, but with the mindset of a true experimentalist. And I want to only focus my discussions on asexual microbes. So of course, there are many communities we can study, but at the starting, we just want to focus on a two-species community of helper and the manufacturer. They can convert, as I told you earlier, from cellulose to a useful product, P. Now, helper, but not the manufacturer, can grow on cellulose. As a helper digests cellulose and grow, it releases a byproduct, which happens to be the sole carbon source for manufacture. And the manufacturer uses a fraction of this growth potential to make this product. And this is the single most important parameter or phenotype in my entire talk, FP, which is a fraction of manufacturer growth used for making P, right? So FP. I carefully chose the parameters so that you can remember them more easily. And the remaining 1 minus FP is used for growth of manufacture. Now, we artificially enclose these, right, with artificial boundaries, such as, for example, in microfluidic droplet or in a microtidal well. We also add a shared resource, such as a nitrogen source, that the two species will compete for. And as you can see, the conversion from cellulose to the product requires simultaneously these two species, and thus, by definition, is a community function. For a community like this, the first question you would ask is that, can the species coexist? That's a very great question, and the answer is no, they may not, right? If the manufacturer spends all its effort making the product, FP equals 1, it cannot grow. But the helper can grow because it has cellulose and a nitrogen source. So by definition, manufacturer would be outcompeted very quickly. However, we can make the FP a moderate and manipulate other parameters such that the two species could coexist. How might it work? It looks like this, right? So initially, the helper can just grow because it has all the resources it needs. Manufacturer initially couldn't grow because it will have to wait for the byproduct from helper to accumulate. So manufacturer will suffer a lag. But if the manufacturer can catch up, that is, grow faster than helper at some point, there's a hope for coexistence. And as they deplete the shared resource, both will cease growth and the product will not be made. In fact, such kind of community has been engineered in the laboratory to make anti-cancer drug. And this has been shown to be stable. By stable, I mean you can start the two species at extreme ratios. It doesn't matter, because the ratios will quickly converge to a steady state value. So this is the community that we're going to study. Now, with the comforting thought that we have a stable community, we can now define a community function. So we started with a newborn community with two types of cells there. And we have cellulose in excess and nitrogen source that's yield limiting. And we allow maturation time T. And then as they grow, the resource will be depleted and the product would accumulate. We define community function as the total amount of product P at maturation time T, or PT. So now there's already considerations that need to be put in for maturation time T. Obviously, you don't want the T to be too long, right? Because otherwise it will take forever to carry out this cycle. You also don't want a very short T, because otherwise you'll be wasting the shared resource. So we have chosen a maturation time T such that most, but not all, resource has been depleted. And this is to avoid the complications of cells getting into stationary phase once they have depleted the nitrogen source. So obviously, community function will depend on FP critically, right? Because if FP is 0, community function is 0. If FP equals 1, this one cannot grow and might even die, so community function is low. So then we have the optimal FP for community function here. So are we ready to do the experiment? Bioengineer in the audience would raise a hand and say, no, let's pre-optimize both species. Maybe we would already be at the optimal. Let's try it. So for helper, what do we want? We want it to grow as fast as it can, right? And accumulate a lot of product to help this to grow. So we can just simply do that by natural selection, allowing them to grow the fastest possible way. Mathematically, we can increase the maximum birth rate or affinity for cellulose and shield resource to its optimal. Indeed, these improve community function. That's great. Now, if we do the same experiment on manufacture monoculture, we would run into trouble. Even though increasing the maximum birth rate and affinity of manufacture for carbon source and a shield resource would indeed improve community function, natural selection will tend to push this FP to 0 because non-producers have fitness advantage over producers. So you get 0 FP, right? So then this won't work. But you could say, I'm smarter than that. I want to do artificial selection on manufacture. What I would do is I would sort the manufacture one each in 96 wells. And I'll give it byproduct, some carbon source, and a shield resource. Now that's already tricky, because how much byproduct are you supposed to put in? Ideally, you want to mimic exactly what's happening in the community. But that's very difficult, right? Because in a community, the byproduct goes dynamically. So you decide, OK, you want to make things simple. You will just add excess amount of carbon of this byproduct. And you put the same amount of resource as if you would give to a community. And you wait for the same amount of time, maturation time T. And then you check at the end of the T, which well has the highest activity. And that is the optimal FP for monoculture. For monoculture. Again, FPs have to be intermediate, because if FP 0, there's no product. If FP equals 1, right, it doesn't grow at all, then it might even die, and the activity, again, is low. But strikingly, the optimal for monoculture production is lower than that is optimal for community. But still, that provides a very good starting point, because starting with 2 pre-adapted strains, one by natural selection, the other by artificial selection, we could fix all other growth phenotypes, such as a maximum birth rate, the affinity for various nutrients. Because if you lower them from an optimal, natural selection will already take care of it. So the only phenotype or parameter that's allowed to change is this FP, which determines, in a heritable fashion, the community function of interest. Natural selection will decrease FP. Now our question is whether community selection would be strong enough to balance, to fight off the natural selection, and thus pushing the FP from this low to high value. So now we are ready to do this computation experiment. We started with newborns. Again, the number of newborns can vary from tens to millions. Obviously, if you have more newborns, you will have more variations to select from. But the experiment is going to get very expensive. So for a starting point, we chose 100 communities to speed up computation. And then each newborn would have about 100 cells, 50 of which is helper and 50 of which manufacture. And actually, the detailed ratio doesn't matter, because the ratio would quickly converge to the steady state ratio, which is around 1 to 1. And we chose 100 cells so that we don't accidentally lose any of the single species. And there could be pre-existing mutations in a manufacture. And again, mutation would only affect one phenotype, which is FP, the fraction of manufacturer growth used to make the product. And we then have maturation time T. As I told you, we chose a T where most, but not all, resource has been depleted. During maturation, new mutations could arise, and changing FP. So the mutation rate is quite high here, but it's not enormously high, because we picked the mutation rate that has been observed in mutators. And this, again, is to speed up computation. Most mutations would be neutral. A small fraction would make FP equal 0. That is no mutation. And another small fraction of mutations will increase or decrease FP by a small amount. And this has been observed when people exhaustively studied JFP mutational landscape. So at the end of maturation, suppose we have a very quick assay that can tell us which is the highest performing community. And then we'll just go for that and split it and allow it to reproduce by generating as many newborn communities as possible, where newborn, the total population size of newborn, is similar to what is from the first cycle. Now, suppose we have depleted the highest performer, then we will go to the second highest performer and repeat this process until we have the same number of newborn communities as the first cycle. Okay, so now let's ask the 3 key elements of selection. First, selection. Here is done artificially. That's no different from artificial selection on individual cells. Second, variations. Mutations, either pre-existing or arising during maturation, would introduce variations in individual genotypes and phenotypes. And these phenotypic differences in FP would cause variations among different communities. But the tricky part is that there are other sources of variations. For example, during community reproduction, each newborn would sample from a fraction of these manufactured genotypes. Furthermore, the total population size and species ratio of the newborns could fluctuate. This means that these newborn communities would look different from each other and also from the original newborn. Furthermore, as a newborn matures, the genotype frequency or species ratio could change very quickly, again, making the newborn of the next cycle very different from newborn from the first cycle. So this would interfere with heredity, which I define as a similarity of a newborn from one cycle to the next. So the question is that, is heredity sufficiently compromised such that community selection would fail? So now let's go to results. So first is a control experiment where we randomly pick communities to reproduce. We performed 3 experiments indicated in 3 different colors. The starting point functioned here, and we see that all of them crashed to 0. That's very understandable, because in the absence of community selection, natural selection would select for FP equals 0, and that would crash the community function to 0. Suppose we apply community selection. So in all 3 experiments, community function fluctuate up and down. They do not crash to 0, but they are no way close to the theoretical optimal as indicated by this purple line. Now, why is that? It's very surprising. So we took a very careful look at one selection cycle and asked how community function correlates with FP, right? Because that's the thing we really want to improve, and that carries heritable information. We actually saw some correlation, but not that impressive. But next, we looked at how community function correlates with total population size of a newborn. And we see beautiful correlation. Community function also correlates beautifully with the fraction of manufacture in newborn. And as we know, these 2 can fluctuate in a random fashion, in a non-heritable random fashion when we reproduce communities. And why is that? Actually, the reason is it's not difficult once we understand what's going on. Remember, I told you that we chose a maturation time where most, but not all resource has been depleted to avoid a stationary phase. And that's for average community size of 100 total cells. So suppose a newborn community happens, by chance, just happens to have a higher population size. What would happen? It would grow to higher final density. It would deplete resource more thoroughly and thus generate more products. Similarly, suppose a newborn happens to have higher fraction of manufacture. What would happen? These manufacturers can't grow, right? Because they have to wait for helper to give them byproduct. So essentially, that introduces a longer than average delay, a lag, before manufacture can take off. So as a result, these communities will not be able to exhaust the nitrogen, shared resource as thoroughly, and as a result, the community function is low. Now, if our hypothesis is correct, then we can improve community selection by extending maturation time slightly, such that, on average, most resource has been depleted. And indeed, that helps. Why does that help? By extending maturation time, we still cannot change the fluctuations in newborn size or fractional manufacture. But you would allow these unlucky newborns that happen to have higher fraction manufacture or lower size to catch up and deplete resource and generate product, just as those lucky ones. So suppose experimentalist says, no, I don't want to extend maturation time because I really don't want to get into stationary phase and endure the complications of sporulation. then you can still do this experiment if you can use flow sorter, for example, to sort a fixed number of cells, and thus reducing this non-heritable variations in community function. And we tested that, indeed, if you fix the total number of cells in newborn, you see that the community function improved toward optimal. And the same trick would also work if the maturation time is a bit longer. And this is actually faster than this. So can we understand the quantitative difference between these two? And I want to introduce you to the concept of community function landscape. Recall that by comparing these two, recall that I told you that community function depends on FP, which is heritable, newborn size, which can fluctuate in a non-heritable fashion, and also fraction of manufacture in newborn, which also can fluctuate in a non-heritable fashion. If we fix newborn size, now the community function would only rely on FP and the fraction of manufacture. So we can plot this in a three-dimensional diagram, where z-axis is community function, and this is the heritable axis, and this is a non-heritable axis. And we get this, this hill. This is a high community function, and that is low. Suppose we start with a selected adult community, as indicated by this magenta solid dot. And we will zoom into this area, and we get this three-dimensional image. And as this selected adult reproduces, it will generate a cloud of newborns. And these newborns would fluctuate in their average FP as they sample from different subsets of genotypes. It will also fluctuate in fraction of manufacture just by chance. And then this is still a bit hard to see, so I'm going to collapse it into two-dimensional picture. This is high function, this is low function. And all the communities on this line would have the equal community function. So here is a landscape for short maturation time. This is the adult community selected to reproduce. It will generate a cloud of newborns. And out of this cloud of newborns, even though these newborns would have the highest average FP, and in fact, we should choose from this, but we are misled. What we would choose is, in fact, this one, because this line is the highest. It's even higher than the highest of this. So we would choose this to reproduce. And as it matures, I added a steady state composition line here. And as it matures, this species composition will be pulled to this green line because of the convergence, this racial convergence phenomenon I told you at the beginning of my talk. So here, note that we have made a very small improvement in FP. Suppose that we extended the maturation time to longer t, then the picture becomes very dramatically different, right? We have this steady state line cutting across these landscape lines. And this community selected to give birth to newborns would have this cloud of newborns. And these will, from here, we will select this one, right? This one would have the highest community function. And as it matures, it will get to here. It will get to here. So we have made a much bigger step of improvement. And suppose here we have this community, hypothetically, that is very lucky in having low fraction of manufacture. It still does not help, because this is still lower than this. So to summarize my talk, I told you a possibility of doing selection of microbial communities based on their function. Besides ensuring species coexistence and pre-optimizing member species in the correct manner to promote community function, although bearing in mind that monoculture optimal may not equal to community optimal, we have to be very wary of large non-heritable variations during community reproduction, and these should be minimized. In our case, these fluctuations are caused by randomness associated with partitioning adult community into newborns. Now, with this community landscape as a backdrop, we can ask many questions that would be of key interest to experimentalists like us. For example, how does the number of communities affect this landscape and the hill climbing of this landscape? And I have told you about the importance of choosing the maturation time judiciously. And then another parameter is the newborn size. Does it matter if a newborn has 100 cells, 1,000 cells, million cells? Does it matter? And the mutation rate. What is the optimal mutation rate you will need for this hill climbing to be the fastest? And the selection strength. Should we select the very top or the top 10% to preserve variation? So all these are incredibly interesting questions that would allow us to gain some insights on general principles that govern community selection. And then this kind of insight can then guide experimentalists to actually perform these experiments. Thank you very much for your attention.
So what I suggest is we can go ahead and set up the panelist table, and if there are any questions, you can start asking them now. But I would request the other speaker also to join us on stage here for the panel discussion.
Yes.
Yes, hi. I'm Sui Wang at ISP. So it's a very interesting model, but you try to minimize the non-genetic variability in the offspring generation, right? But according to newer theories of Darwinian evolution, I think that's also part of evolution, and that could accelerate.
Actually, no,
right?
I cannot hear what you are saying because this one is going up.
So
now I can hear.
I was asking about the non-genetic variability
That's
of
right.
your every generation, and whether that actually is a essential element in evolution. That people talk about, genomic accommodation, genetic plasticity, and so on.
That's
Do you
right.
use that?
So I think in this scenario, it hinders our ability for this particular scenario, where we just partition an adult community into newborns, because newborn size can fluctuate right, in Poissonian fashion, and the ratio can fluctuate. These introduce non-heritable variations in community function that hinders our ability to select for high FP, because you can have a community that has very, very good average FP, but because they're unlucky, they happen to have smaller total number of cells or unfavorable ratio. So they end up not seeming to be that productive, and the experimentalists will then not pick it. And there could be the counter case where the FP is actually low because they have no non-producing mutants. Right? These ones will actually quickly become very bad in the next cycle because the non-producers will quickly rise in frequency. But these can fool us into thinking they have high function because they happen to have more cells to start with. And these fluctuations you can understand if you just, as experimentalists, you take a pipette, you pick up 10 microliters, right? That 10 microliters, even if it gives you average of 100 cells, you could be sampling 120 or 8 days. But these difference in this kind of simulations actually makes a huge difference. So that is these sources. And then there could be other sources, right? The measurement precision that can also introduce problems, right, when you are selecting these communities. So